आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
We bring you all a podcast series on STEM popularization, that is, science, technology, engineering, mathematics, and medicine, supported and catalyzed by the National Council for Science and Technology Communication, the Government of India. Program is produced by Media for Community Foundation.
Welcome to the podcast from Indo French Center for the Promotion of Advanced Research, or CEFIPRA. We are here to showcase the exciting collaborations between Indian and French scientists, spanning a wide range of scientific fields from fundamental research to applied sciences. Now, let's listen to them.
Hello everybody, my name is Dr. Himanshu Sinha from Department of Biotechnology, Bhupat and Jyoti Mehta School of Biosciences, IIT Madras. I work in the area of yeast population genetics, and my main interest is to understand genotype-phenotype relationships between how the phenotype changes as the environment changes, as well as the genotype changes. So this allows us to understand how various phenotypes evolve over time and what phenotypes are adapted and what phenotypes are selected as the population evolves. So in this current CEFIPRA project, which is with Dr. Gianni Litti from University of Côte d'Azur in Nice, we are trying to understand how this particular relationship between genotype and phenotype is in context of antifungal drug resistance. So the lab of Professor Litti has sequenced and phenotyped about 1,000 diverse population of yeast strains, which has been acquired from variety of sources, including both environmental, clinical, human sources, as well as brewing and baking. And using this population, what we want to achieve is to understand if fungal drug resistance occurs, what are its natural sources, are there any natural variants that already exist in nature which allow the yeast to acquire drug resistance, whether there is any specificity of drug resistance variants in populations that have been derived from human sources versus that have been derived from environmental sources. And this would also allow us to therefore figure out whether there are natural reservoirs of antifungal drug resistance which yeast strains can quickly evolve to. So this is basically the project, and we have been working on this for past 2 years, but because of COVID lockdowns, the labs in both the sites, both here in Chennai as well as in Nice, were in different phases of lockdown, so the progress has been slightly delayed. What we are very interested in is to understand how antifungal drug resistance evolves in natural populations. For this, what we did, we phenotyped all the 1,000 yeast strains in the collection for a variety of antifungal drugs such as azoles, etc. And based on the phenotype and the available genotype information of all these 1,000 strains, we perform genome-wide association studies to map various variants that give resistance to these antifungal drugs. One of the other areas of focus is to understand whether these drug resistances are under any selective pressure, especially when these fungi are infecting humans, because when a particular yeast infection happens, drugs are not the only resistance that they have to overcome. They are also have to, these strains also have to overcome oxidative stress resistance versus genotoxic stress resistance, and therefore what we are also doing is evolving some of these candidate strains in multiple different drugs, multiple different stress conditions, both alone as well as in combinations, to understand what are the evolutionary adaptations that these strains undergo to overcome either one stress or a combination stress. And this would allow us to get a more comprehensive understanding of how drug resistance develops in fungi. On the other hand, for the evolution studies, what we want to do is to now sequence these evolved strains, because these evolved strains have been evolved either in drug alone or drug with particular combination, stress combinations such as oxidative stress, and try to compare the molecular mechanisms, the mutations that have been acquired in these evolved strains, and therefore compare molecular mechanisms that are present in the strains where the strain was only evolved in the drug versus when the strain was evolved in drug plus oxidative stress combination, for example. This would give us a better handle on how resistances develop and whether there is a trade-off in developing a resistance against two opposing factors. One is the antifungal drug resistance, other is oxidative stress resistance. And this is something that has not yet been done in most of the evolution studies, because most of the evolution studies have only studied how drug resistance is acquired, which is a very well studied area of research across different kinds of fungal pathogens.
This episode is brought to you with the content support from Cefipra and the technical support from the Sparrows team. We extend our heartfelt thanks to the Director of Cefipra, Professor Nitin Seth and Dr. Payal, for their continuous guidance and support. Stay tuned for more episodes and thank you for joining.
आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी